Okay, why don't we get started? This past week in lab, you guys collected data for the neuroanatomical part of this module, and next week we'll be analyzing that data, looking for differences in hippocampal volume between males and females and between younger and older subjects, and right-left differences. So we'll be doing the analysis on the data next week from the data that you collected. And what I wanted to do in lecture today is go into a little more detail about the anatomy of the medial temporal lobe circuit, which you're already familiar with, and certainly you're pretty good at seeing the hippocampus in the brain. And we'll talk a little bit more about some of the details about that anatomy, and particularly a model that Eichenbaum and Lipton developed of how the hippocampus gives rise to episodic memory based on that anatomy. And then we'll also talk about measuring episodic memory in animal models. The way that I defined it last week and the way that we're going to be using it in class when you do the memory test is really based on introspection. Are you aware of reliving that moment you saw an item? Are you aware of reliving that event? In animals, that's not so easy to do. They don't have verbal abilities. We can't ask them things, but how have people tried to get at this question in animal models? How possible is it? So we'll talk a little bit about that and try to make some links between the human work, which is what our focus is on in this module, and some of the literature out there on animal models of episodic memory. Okay, so medial temporal lobe anatomy. Last week I introduced this, that the hippocampus lies at the top of a hierarchically interconnected loop. Here are the hippocampal regions here, getting inputs from entorhinal cortex, which got inputs from the parahippocampal and perirhinal cortex, which got inputs from all over the brain, from association cortices and all the different lobes. And in addition, there's feedback through entorhinal cortex and back. So it's like a loop. So we went over that last week. And just a little bit more about the anatomy. Here you see a human brain from the center looking out. And here are some of these regions we've been talking about. So these are cortical regions. Here's the parahippocampal gyrus. And two other gyri you might have heard of. The lingual gyrus back here by the occipital lobe. And has anyone heard of the fusiform gyrus? That's a big major gyrus in the ventral part of the temporal lobe. And it includes regions that are important for high-level vision, like you might have heard of the fusiform face area, that you find regions in the fusiform area that seem to be particular to faces or complex visual stimuli. And from this medial view here, you see the parahippocampal gyrus. This is the cortical area that is the cortical gyrus that is covering up the hippocampus. The hippocampus you can't see from the medial view. It's all hidden in here. Okay, so this view you're probably extremely familiar with after drawing all those sections. So in those brain sections that you saw were sliced kind of like this. A coronal section, and they looked like this. And here was your hippocampus that you drew. And areas that you learned to avoid when you were drawing that is this part here, the entorhinal. Oh, actually, here, this is a blown-up view. You were drawing the hippocampus. You avoided the entorhinal and perirhinal and parahippocampal cortex, these cortical regions. So in the anterior part of the medial temporal lobe, so here would be one of those more anterior sections that you guys were looking at. A little bit more anterior. You'd see the hippocampus and the entorhinal cortex and perirhinal. As you went back, and when the hippocampus started to look a little different in the more posterior caudal sections, the entorhinal and perirhinal cortex weren't there anymore. The parahippocampal cortex extends much further caudally. So the parahippocampal cortex is the caudal part of the parahippocampal gyrus. The parahippocampal gyrus includes all these other cortical areas. Parahippocampal cortex is the part that extends back the furthest. Okay? So these areas you probably saw in all your sections, and you made your decision to stop circling about there usually. Okay. So how does this relate to other animals? So I've been really focusing on primate anatomy in this module. The brains you were looking at were human brains, and the anatomy that I've been talking about was developed through research with macaques, with non-human primates. But how does this relate to work with, say, other animals that are more tractable in a lot of experimental kinds of paradigms? So if we look in the rat, of course, rats have a hippocampus. They have a lot, it's fairly similar neuroanatomy, but the cortical connections are less developed than in the primate. So in the rat, some of the major projections that you've probably heard about are these subcortical projections, like the fimbria fornix, that connections to the mammillary bodies, connections to the septum. Those are really the ones that you really focus on, and often just cutting the fimbria fornix in a rat is almost as good as taking out the hippocampus in terms of the kinds of effects on memory it has. In the primate, I would say that the projections to cortex are more dominant. That a fimbria fornix lesion in a human, though I wouldn't say that it doesn't do anything, it's not as bad as, say, a lesion of hippocampus or a lesion of the entorhinal cortex or any of these cortical regions. So it seems like in primates the cortex has a bigger role. And that shouldn't be surprising. The cortex is much more developed in primates than it is in rats. We have evolved much more extensive cortices. It makes sense that they're doing a bigger role in memory processing in primates than in rats. However, in rats, you do see these kind of cortical connections. In rats, you don't see a parahippocampal gyrus the way that I pointed out to you in the primate brain, or you saw in your human brains. But you do see these cortical areas. So you probably have all seen a rat brain, and they're nice and smooth like this. They're not all these sulci and gyri like in the primate brain. They have a lot less cortex. But you can, by doing anatomical connections from the hippocampus out and from the different areas, you can define cortical regions on this smooth brain that do have some of those similar projections. So you can find the entorhinal cortex in the rat looking at regions where the hippocampus, where you have projections from the hippocampus and projections into the hippocampus. And also, there's a perirhinal cortex that includes Brodmann's areas 35 and 36, and a parahippocampal cortex. So there does seem to be these cortical areas that are kind of in this part of the rat brain. So here's in orange the entorhinal cortex, and this yellow is like the primate parahippocampal cortex, and this green and red are areas that are kind of analogous to the primate perirhinal cortex. And see how the perirhinal cortex-like areas are more rostral, and the postrhinal or parahippocampal cortex is more caudal. Okay, so in the rat there's some analogy. You don't see a nice gyrus like you do in the primate, but there are areas, if you just base it on anatomical connections, that look very analogous and also seem to have some functions in memory. Okay. Question? Oh, okay. So you guys have heard of Brodmann areas? They're areas, you can take the cortex and define specific areas based on the shapes of the cells there. And so even if you don't know anything about function, you can just say, oh, this area looks a little different than that area. And Brodmann named all those areas. And so two areas, area 35 and 36, so they have somewhat different cell structure, but they're both projecting into the entorhinal cortex. So they seem to be analogous to the primate perirhinal cortex. Okay, so what in the Eichenbaum and Lipton article, a lot of it is based on this idea that maybe the perirhinal and the parahippocampal cortex have somewhat different roles in memory. They're both feeding up into the hippocampus. Maybe they're doing somewhat different things. They're not just kind of replicating each other. And just to understand their view, you have to know that there are two different major cortical streams in the brain. And it's been most studied in the primate brain, but would be true with other brains as well, other animals as well. The idea that there's a dorsal and ventral stream coming from the occipital lobe, coming from V1. Is this idea kind of familiar that you might have heard in other classes? Yeah. So this is the dorsal stream goes from occipital lobe up into the superior parietal lobe. So it are connections to areas into the parietal lobe. And the ventral stream is here in the temporal lobe. So like into the fusiform gyrus, for instance. Okay, so there are these two major pathways in the brain. Okay, and they've been studied extensively in primates who are very visual, and so these are major projections. Okay, so in the dorsal and ventral streams, they've been divided into having two different roles in telling us about the world. So why do you have a visual system? Well, one thing is to tell you what things are based on vision, and another thing that your visual system has to do for you is tell you how to interact with items. So the idea is that these two streams fulfill these two different roles. The dorsal stream is more telling you how to interact with something. If I want to pick up this pointer, I need to tell my motor system its location, its shape, and program my motor system in order to do that. However, if I have a few different items and I want to identify them, my ventral stream is going to do that kind of information processing. So dorsal pathway, I would call the how pathway. In some classes, you might have heard it referred to as the where pathway. I think that how is a little bit more accurate because it really describes what it's doing for you. It's telling you how to interact with items. Of course, their location is a big part of that, but I think how captures it a little bit more accurately, and I think that reflects more the current view in the field. And the what pathway is telling you all about particular items, what they are, helping you identify them. And they terminate in these different areas. One is more parietal, and one is in the inferior temporal lobe. Okay, now if you look at the parietal and parahippocampal cortex, it seems that they get different amounts of input from cortical areas in these two different streams. So the parietal cortex receives input primarily from the ventral stream, so from the inferior temporal cortex, whereas the parahippocampal cortex receives input from the dorsal stream. It's receiving a lot of parietal information. So just based on that knowledge alone, just looking at its connections from these different cortical areas, you may think that, well, these are having different roles. These might be the kind of dorsal and ventral stream inputs into memory. So it's kind of like this segregation is being preserved at the level of what's going to feed into the hippocampus. Okay, and then furthermore, as you saw on that diagram, both the parietal and the parahippocampal cortex are feeding in to the entorhinal cortex. So you might say, well, everything's going to get mushed together at that level. Well, actually not. If you go look at the anatomy in a little bit more detail, you see that that segregation is even preserved at the level of the entorhinal cortex. And that's one of the points that Eichenbaum and Lipton make. There's segregation of the inputs into the entorhinal cortex. The parietal cortex will mostly go to the lateral entorhinal cortex, and the parahippocampal cortex mostly goes to the medial part. So even at the level of the entorhinal cortex, these things are segregated, and they really don't get integrated until the hippocampus. Okay. Now, what's the evidence that the parietal and the parahippocampal cortex are processing two different kinds of information? Well, a couple of lines of evidence. Just recordings from parietal neurons in monkeys mostly show that these neurons respond to objects. So kind of like neurons in the ventral stream, they will respond. Maybe there'll be a neuron that responds to something shaped like this, but not a coffee cup, and you have another neuron responding to some other visual stimulus, or it'll respond maybe to a blue coffee cup but not a red one. So it's very specific to particular items. And you see that in the ventral stream, and you see that in perirhinal cortex too. And one thing about these perirhinal neurons is they definitely modify firing based on how familiar these objects are. You show the perirhinal or the subject a stimulus, you're recording from a neuron in the perirhinal cortex. The second time you show the stimulus, you almost always get a big decrement in firing. So it seems like it cares about familiarity, or you could say that it likes novelty is kind of another way to think about it. A novel object will often fire these neurons more. Subsequent presentations, you'll have a decrease. So you can see here how it is a way to kind of measure familiarity, or a familiarity signal. A reduction in firing is equated with, oh yeah, that looks familiar. Now, in the parahippocampal cortex, you don't really see so many of those kinds of neurons, but there's a lot of evidence, particularly from fMRI experiments, that in the parahippocampal cortex you see a lot of spatially activated regions. So in experiments where you're, say, measuring bold signal, measure of blood flow into the brain regions, you find that in the parahippocampal cortex, you can usually see what's called a parahippocampal place area, a region where you get increased signal with, when the subject is looking at spatial locations and not objects. Even objects that are equally complicated or colorful or interesting, those kind of objects don't activate this area. It really needs to be a location. And the kind of location that it can be is quite varied. So you can see this area activated pretty much equally for things like nice outdoor scenes, but as well things that are more man-made, not natural scenes, but like this building, and even interior scenes. So you show them pictures of living rooms or kitchens, it will equally activate. So even though these pictures are pretty different in terms of their meaning and what kind of location, they can all equally activate this parahippocampal place area, whereas just showing a couch or showing a tree will, like this one, will not activate this region. So this is consistent with the idea that this, and you don't see this kind of activation in the perirhinal cortex. It's really, which would not discriminate between, it would maybe like this picture, but it would like the tree just as much, a particular cell or a particular region. That region doesn't really care about is it in a location or not, just likes objects. Whereas parahippocampal place area within the parahippocampal cortex seems to be like, I want this in a place. I don't care about objects, I want a place. Okay, so a lot of evidence that these two regions are processing information differently. Okay. So based on these kind of data, Eichenbaum and Lipton draw them kind of together and develop a theory based on this information. So what they posit is that when you recognize an item, so an item being, say, next week when you take that memory test and you recognize one of the words, or you see somebody in Ralph's and you say, oh, I know that person, or an object that you've seen before, anything like that. When you recognize an item, the idea is that you can activate the perirhinal cortex and thus the lateral entorhinal cortex. And if you activate those regions, that gives rise to this feeling of familiarity. When you see somebody in Ralph's and you're like, oh, I know that person, I'm sure I've seen him before, that's because you're activating these regions, perirhinal and lateral entorhinal. And activation of those regions is adequate to support that kind of feeling of familiarity. But of course, we made a distinction between that kind of feeling and real episodic memory. Now, when you remember the spatial and temporal context of something, which is key for episodic memory, when you actually remember, oh, I see that person, and I remember yesterday I had this conversation with them. In that case, to have that memory for that place and the event, you have additionally activated the parahippocampal cortex, which is all about places. And in Eichenbaum and Lipton, they also argue, well, it's probably about temporal context too. In that case, you're activating parahippocampal cortex and the medial part of the entorhinal. So you're getting this additional activation if you have both of those things going on. And when you have all that activation, you're going to activate hippocampus and have one of these more episodic memories. So in the memory test that we'll give you next week, there'll be some items where you actually can sort of put yourself back in time and remember seeing that word on the screen. And that's because you're activating not just your perirhinal cortex, but also your parahippocampal cortex, and you're activating both regions of the entorhinal and thus driving the hippocampus. Yeah, question?
Since when you recognize an item, you may activate the perirhinal cortex. With other perirhinal, the more familiar it is, the less it fires.
Yeah, but I think that the case is that a reduction in firing is still information, because you can imagine that it is, if it's reducing firing, then regions downstream may be disinhibited. So I think that and it's also inhibitory firing increases bold signal. So I think it's the case that you can't just say, well, more action potentials mean more information, that actually a reduction in a baseline could be very important information that these structures are using. So in fact, if you record neurons in the hippocampus, when subjects remember items, what you often see is a lot of inhibitory firing. And there's a lot of theories of why that could be. Is it because you want to increase the signal to noise for the few neurons that are encoding it, or actually the reduction in firing is the information? I mean, anytime you have a change, I think that's valuable information. So even if you're having a reduction in one region, that could be translating into changes in downstream regions. But you do have a really good point that we still don't fully understand the circuit properties here, and there are computational models trying to model how these changes in firing could alter regions downstream. So that's kind of part of ongoing science, how we can take what we know about what the cell firing does and put it into a model that we can actually get memory out of. Other questions? Yeah?
From last lecture, just connecting it, is the parahippocampal more dependent on the amygdala's function? Or?
I don't think necessarily. Now, I would say, if anything, there are more projections from amygdala to perirhinal, but there are certainly projections in both regions. So there are, the amygdala does project to the medial temporal cortex, but I don't know if necessarily one region is really getting the dominant input. I just think there may be more into perirhinal just because it's closer. The amygdala is really kind of wrapped around, or the perirhinal is kind of wrapped around amygdala, and that's a fairly strong projection there. But it really is projected to all these areas. And so that kind of boosting of memory you get with more emotion, I don't know if that only happens with its projections into perirhinal. It's probably happening in the entire medial temporal lobe. Okay, so that's really the Eichenbaum and Lipton model that really they are endorsing the idea that to get a real episodic memory, you're activating hippocampus. And when you have both the item and its spatial temporal context, you get enough changes so hippocampus is actually being affected. Okay, so they would say that, let's say you have a really vivid memory for an item. They say that, well, at that point, maybe you get strong enough activation of the perirhinal and lateral medial entorhinal cortex to activate the hippocampus. Because remember, that goes in and drives hippocampus, so causing changes in hippocampus. And as you know, there's these back projections out into the entorhinal and parahippocampal cortex. And so perhaps if you see someone or something that you have a very vivid sense of familiarity for, and you really recognize that item, that can kind of, through the back door, through the hippocampus, go back and activate the parahippocampal cortex and the medial entorhinal cortex and give you that kind of full-blown episodic memory. So it could either happen when both things are activated at the same time, the context as well as the item, or it could happen because the item is so vivid and strong, it itself can activate hippocampus, which can go back and activate the parahippocampal cortex. So the Eichenbaum and Lipton article is basically taking that neuroanatomy and behavioral work suggesting that the hippocampus is important for episodic memory and kind of trying to form an idea of how it could work. Okay. Any questions? No. Okay. So the other thing I wanted to do today is talk a little bit about episodic memory in animal models. And there's a bit of a challenge here. So in this module, I've really been stressing this kind of almost introspective idea of episodic memory, that it's like, oh, when you feel you're reliving that moment, and on next week's memory test, we will be emphasizing that when you're trying to decide whether this item was a R or a K, a remember episodic memory, or just a no, a feeling of familiarity. So you're really going to have to kind of make decisions about your own memory, kind of what's called meta-memory decisions. And so it really depends on this kind of conscious awareness. Now, the question is, well, do animals have that? Do they actually have this kind of feeling of going back in time and reliving an event, or do they just sort of act, in the moment? And, I think it's very difficult to know, if not impossible, if animals are more kind of living in the moment or if they're actually able to relive events the way that we can. So kind of the difficulty is that episodic memory is this kind of characterized by mental time travel. That's how I've been describing it last week. You mentally re-experience the event. And what about non-human animals? Do they even really have this experience? They certainly have the machinery in terms of this medial temporal lobe that kind of looks like other primates, certainly in terms of its connections. Even in rats, you see quite a lot of analogy. But do they really have this same experience? How do we test this? Because we can't really ask them the way that I'll be asking you next week when we collect our data on episodic memory. So some very interesting work has been done on this using this species, the scrub jay, western scrub jays, which is a species of blue jay that you see it in California, I think mostly northern California. Like if you go up to the Sierras, you'll see a lot of these very nice birds. Pretty loud, I guess, but they're pretty. They're nice blue color. Western scrub jays. And one thing about these animals is in their natural habitat, they're expert food cachers. This is true with a lot of birds, but these are one example of a bird that is really good at this. They will take food and they'll hide it in a ton of different places. Now this makes sense for an animal that lives in a climate where there may be one period of time where there's lots of food around, and then another period of time, like the winter when it's snowy, where there really isn't food. And so what that animal needs to do is go back to where it has cached the food. It's like hiding the food, and so they don't starve over the winter. So they've evolved these mechanisms to hide their food and remember those locations. And it is actually quite amazing. They can remember tons and tons of these locations. So they do seem to have spatial memory in that they can remember 100 locations to get them through the long winter months when food isn't prevalent. And in some experiments that are done in the laboratory, there's some evidence that suggests that not only do they remember the locations where they've hidden food, but actually the time. So now this is starting, if they can remember the time and place of an event, that is the time and place they've hid food, that's starting to sound like an episodic memory. So let me describe some of the experiments done with these animals. So scrub jays eat both peanuts and worms, eat a lot of different things, but of these two types of food, if given the choice, they'd rather have a worm than a peanut. That's just how birds are. I mean, I guess there's more, these are tastier to a bird than a peanut. They'll certainly eat peanuts, and they'd rather eat peanuts than starve. But if you give them a choice of one or the other, they'll go for the worm every time. Okay, so worms are preferred food for birds. Okay, now in these studies by Clayton and Dickinson, these birds were given some of each food type and then allowed to cache the food. So if you give birds, a big pile of these two kinds of food, they'll maybe eat some, but they will always take it and any excess and take it and hide it wherever they can. That's just part of their behavioral repertoire. They're cachers. They want to hide that for later. Okay. So in these studies, they were able to have an environment where, show on the next slide, where they had these ice cube trays filled with sand, and with a pile of food, they will hide the worms and peanuts in these ice cube trays. And so the experimenter can make a note of which place they hid the food. And they usually have, here they're using Legos, some kind of spatial information so the animal can tell, which side is which, to give some context to the room. Question?
Are these bunches raised or from the wild?
I believe these are captive raised and hatched, that they're not just caught in the wild. Nevertheless, they still show this food caching behavior. They don't have to have lived out in the Sierras in order to do that. Even birds reared in captivity will show this kind of behavior. Okay, so they're caching the food, and then what the experimenters can do is bring them back later and put them in the same environment, and here they will clean out the tray because they don't want the animal, when they want to look to see where the animal is looking for the food, they don't want the animal to have any other possible cues available like smell or sight or marking it somehow. They actually put in a new cleaned ice cube tray with brand new sand with actually no food in there, and they just measure where the blue jay goes in order to see where its memory is for that particular food. Okay, so that would be the test. So let him cache, have some delay, bring him back, and see where he goes for the food. So the findings are, so after he's hidden a bunch of worms, a bunch of peanuts, after a short delay, these jays went for the places they hid worms. So if you brought them back, say within a day, they would first go to the worm hiding places that they personally hid worms. However, if you brought them back over many days, you gave them a big long delay, they preferentially went for the places they hid peanuts. Remember, there's no peanuts or worms here because we don't want to give any cues, but they go back to those locations where they had hid the peanuts. Why? Even though, of course, they'd rather have a worm than a peanut at any given day, presumably because the worms would have spoiled. If you wait a couple of weeks with a dead worm buried in the sand, it will not be so tasty, even if you love worms, compared to a peanut, which can last really a huge long time. So it seemed that these data suggest that the jays are remembering the time and place they hid the food. So the idea is the jays put back there saying, okay, let's see, I was in here a couple weeks ago, so let's see, I remember hiding the worm here. I don't want to go for that. Oh, I hid a peanut over here. Whereas if after the short delay within a day, they're actually remembering, okay, I remember hiding the worm here, I'll go for it. So it's not that the animal is just saying, oh, I know some locations that I like food. They're actually remembering when they hid something. Okay. Any questions? Okay. So this has been kind of a challenge to the idea that, well, episodic memory, big part of its definition is this sort of experience of reliving it. We can't ever get at that in animals, but it suggests that, well, maybe that shouldn't be part of it, that maybe if we just have some way of making sure that the animal needs to know the time and place of something, that is a good definition of episodic memory. But then the question is, well, maybe animals are experiencing it the same way as us. So maybe that blue jay is, just like how I said, really re-experiencing the moment that he hid the different foods in order to solve the task in the way he did. However, there is some controversy about that. It's not because we can't ask them, and it's not really completely clear if the Jays are really recollecting the moment they hid the worm. I just said that they are kind of consciously reliving that event, but others would argue, well, if you actually could ask the Jays what they're doing, they may not have that time travel reliving aspect of the event. They may be just saying, well, I just knew, for some reason I just wanted to go to these locations. I don't know why. And then if you had that long delay, I don't know why, I just wanted to go to these locations. We don't really know, and it's not really knowable. So that is a controversy in the field now. What is really the essential part of episodic memory? Is it that kind of conscious re-experiencing, or is it just the fact that you have spatial temporal context, which you could argue these blue jays do? But whether they have that kind of conscious awareness feeling that I think is a big part of what we think of in episodic memory in humans, what makes it kind of cool and special and, part of our personal past, is that really a key component in what the hippocampus is doing for you, or not? And I think that is one of the big issues that we're grappling with. We don't have good answers to. But certainly people who work with animal models are really trying to the proponent of this idea that, well, actually maybe it's not, the conscious aspect of it isn't as important as the spatial temporal aspect, whereas I think the human researchers are very much tied to this idea that this re-experiencing, this kind of conscious re-experiencing of an event is a really big, important part of this type of memory. Okay, any questions about the Jays or episodic memory or what we did in lab this week? Okay, well that's it for today, and next week we will analyze the data, we'll give you the memory test, and for those of you who weren't able to finish drawing your cerebrums, you'll have plenty of time to do that also, because we'll make sure we have all that data collected by next week.